Hello, welcome in this video lecture. In this lecture, we are going to study structures of protein, four different levels of protein structure, that is primary structures to quaternary structures, and lastly the properties of protein. So let's we start. What is the structure of protein? So we have already discussed, that is amino acids are the building blocks of protein, and proteins are built by linking of two or more alpha amino acids together in various orientation and configuration. So as the alpha amino acids are joined together to form peptide bond by condensation reaction between two alpha amino acids, it releases protein as a peptide bond formation, new molecule, and water is eliminated. So very easy reaction. During this linkage, we see how the peptide bond is formed. That is, one of the COH group from first alpha amino acid combines with NH2 group of second alpha amino acid, either same or different, and it gives reaction between carboxylic acid group and NH2 group so as to form new peptide linkage, and in this manner new peptide bond is formed and water molecule is released. This is simple condensation reaction. So whenever different peptides are formed by the arrangement of alpha amino acids and alpha amino acids by different combinations, various kinds of proteins are developed, and these proteins are having different in their size, structure, and functions. Now how this combination takes place, see the below first point, bullet ने दाखवलेला आहे. Bullet, see the first point. Peptides are of 2 to 50 amino acids. Remember, minimum 2, maximum 50 alpha amino acids are combined, condensed together so as to form number of peptide linkage, and they are classified as oligopeptides or polypeptides. See, oligopeptides or polypeptides means the proteins or peptides composed from 2 to 50 different alpha amino acids, and the polypeptide is a chain of 20 or more alpha amino acids. So according to this, various alpha amino acids combined and having limitations of 20, 30, 40, 50 different alpha amino acids are simply composed as the peptide bond, and such type of peptide linkages in the protein. Such proteins are also called as oligopeptides. So simply oligopeptides are composed from 20 to 30 alpha amino acids with their some residual combination. So this is the first combination. Second, peptides mainly have a linear structure, four structural levels, that is primary, secondary, tertiary, and quaternary. So according to this, the most of the proteins are having the arrangement, their size, their structures and functions, and when their proteins and their structures are considered, there four types of protein structures are There, that is four levels of protein structure. Out of these four levels, how they are classified? These are classified according to the arrangement of these particular amino acids present in the protein in different manner. Structure, whether they are linear structure, whether they are having a spherical structure, whether they are having twisted structure. According to this, there are four levels of protein structure, and let's we start which are these four levels. Now see in the particular nature. See the first one, that is the four in out of four there is this is the first structure, that is known as the primary structure of protein. Now see the primary structure of protein, how it is developed. In the primary structure, you are looking in the diagram. This is various types of alpha amino acids are shown here. So in primary structure, there is arrangement of alpha amino acids into linear peptide chain. See the first structure, linear peptide chain, this one A chain. A chain is the linear combinations of various alpha amino acids, and this is linear polypeptide chain is developed. Observe the chain A starting from GLY stands for glycine, ILE stands for isoleic acid, valine, GLU stands for glutamine, GLN glycine, CYS, CYS stands for cysteine, and again CYS stands for another cysteine. So GLY short form of glycine, ILE isoleic acid, VAL valine, GLU that is glutamine, GLN is the glycine, CYS is the cysteine. All these are the short form or codes of alpha amino acid, and whenever see the A chain, in the A chain these particular alpha amino acids are linearly combined, so this protein or peptide is called as primary structure of protein. So very simple. Primary, in the B chain there is a linear nature and later on it is turned. Linear nature, later on it is turned. So PHE, see the B chain. B chain is nothing but it is the PHE. PH is the phenylalanine, valine, ASN, GIN, histidine, lutein, cysteine, glycine, serine, histidine, lutein. So all these are the different alpha amino acids combined in the linear manner, and this is the primary structure of alpha amino acid. Then second is the secondary structure. In the secondary structure, I will show first you the diagram. Secondary structures are having two arrangement. In the secondary structure, there are having two arrangement. Focus on diagram. What is the diagram? One is the alpha helix and another one is the beta pleated sheet. Both are different. See, in alpha helix, this is helical structure like spring, and how it is developed. See the alpha helix structure in the bonded form, that is bond formation takes place between hydrogen of NH group of one alpha amino acid with carbonyl group of second alpha amino acid. Similarly, NH group of second alpha amino acid combined with carbonyl oxygen of third alpha amino acid. This is secondary protein structure in the form of alpha helix. Now see the beta pleated structure. In the beta pleated structure, it is similar to linear but in zigzag manner. but in zigzag manner. So beta plated structure and alpha helical structure comes under the secondary structures. Now see the definition of alpha helix. In alpha helix structural form, carbonyl group of one alpha amino acid is hydrogen bonded to alpha amino acid of second hydrogen atom of second alpha amino acid of the fourth amino acid in the sequence. So hydrogen bonding takes place in this manner, then it is alpha helix. Okay, see shortly. In alpha helical structure, carbonyl group of one amino acid is hydrogen bonded to amino hydrogen of fourth amino acid in the sequence. This pull the linear chain into helical ribbon form. This is very important. Linear structure is get converted into alpha helical form, and each helical turn contains 3:6 amino acid, 3 to 6 alpha amino acid. So this is the structure, secondary structure of protein known as alpha helix. Now see, again I will show how the alpha helix takes place. See, alpha helix, first to fourth, linear structure is converted into alpha helix. Now see the beta plated structure, beta seed or strand. Two or more segments of polypeptide chain are linearly up next each other, and strands are hydrogen bonded with each parallel or antiparallel form. It is called as beta plated structure. This is the secondary protein structure. Now in the tertiary, see the tertiary structure. See the slide. In tertiary structure, what happens? The polypeptide chain is further folded into three-dimensional space. It is different than primary structure, linear structure. Secondary structure of alpha helix and beta splitted. In tertiary structure, polypeptides are formed into three-dimensional space involving one or several domains. In alpha helix and beta plated structure, the folding is globular, while in tertiary structure it is having a polar and non-polar type of bonding, and there is a spherical or three-dimensional space is developed. Such kind of structure is known as tertiary structure. So this is about the tertiary structure, and next one is the quaternary. Now how the quaternary structure is developed. See the fourth level, quaternary structure. This quaternary structure is like this. Fourth one, see the structure. Quaternary protein structure consists of more than one alpha amino acid, and it is combination of so many globular, alpha helical and pleated sheet in combination with each other, known as quaternary protein structure. So these are the four different structures which are present in the protein. Primary proteins, see the diagram, it is linear. Secondary, alpha helical and beta splitted. In the tertiary, there is a particular space is given in the form of ribbon, and fourth one, it is combination of the primary, secondary and tertiary in the form of globular structure. So these are the four different structures observed in particularly the various kinds of proteins. So in this short lecture we are going to stop here. Next properties we have see in next video lecture. Okay, thank you.